Okay, so thank you so much for joining us. My name is Alyssa Noor, and I'm a current Cancer Biomedicine student here at UCL, and I'll be your chair for this afternoon's session. So I'm hoping this session will address those course-specific questions you may have and will help you to gain an insight into what it is really like to study on our Cancer Biomedicine undergraduate program. So this afternoon, our speakers are Dr. Ursula McGovern and Dr. Alwyn Dart, and we'll introduce you to some of the great science and medicine that you would learn as a student of the Cancer Biomedicine BSc and MSci here at UCL. So our speakers will also give us a summary of the Biomedicine program, and then there will be a short Q&A session at the end for the second part of the event. Um, no worries, if for the people that are still joining, the session is recorded and we'll also make it available on the website following today's event. And we're here to respond to your questions, so please share those throughout the Q&A function on the Zoom. So before we begin, let me introduce your speakers. So we have Dr. Ursula McGovern. So Dr. Ursula McGovern is an honorary associate professor at UCL and a consultant medical oncologist at the University College London hospitals. So she specializes in prostate and bladder cancers, and she has a PhD in molecular oncology from Imperial College London, and is also actively involved in clinical research. This is particularly in leading trials for advanced urological cancers alongside contributing to undergraduate oncology teaching at UCL. And we also have Dr. Alan Dart. So Dr. Alan Dart is a lecturer and research scientist with over 20 years of experience in cancer research, specializing in prostate cancer. And his work focuses on understanding disease progression and treatment resistance. So this is with the aim of developing new therapies and enhancing the effectiveness of existing ones. Dr. Dart also investigates the role of androgen receptors in normal physiological systems and contributing to broader insights into cancer biology and therapeutic innovation. And lastly, also joining us is Dr. Thiros Rantharan. So Dr. Thiros Rantharan is an associate professor and program director at the UCL Cancer Institute, and he's an experienced academic in biomedical science, teaching across undergraduate and postgraduate levels. So his research focuses on host pathogen interactions, viral carcinogenesis, and novel anti-cancer and antimicrobial agents. So since September 2024, he has also served as a student success lead. And Dr. Srantharan will be online throughout today's event to help support any questions that you may have. So over to you, Dr. McGovern and Dr. Dart. Thank you very much.
Thank you, Alyssa. So, welcome everyone. It's lovely to see so many of you online with us. So, I'm going to start by just sort of setting the scene a little bit, talking about cancer in 2026, and then letting you know a little bit about the course. So, why is cancer biomedicine important? Why do we think it's so relevant? So, cancer is very common, and we feel that, really whatever sort of field within the biomedical sciences you're working in is a very relevant disease process. And if we think about cancer, often now as a chronic disease, there's a lot of similarities between other conditions such as diabetes and airways disease. So we think it's a very relevant topic to be thinking about if you're thinking about a career choice and a degree choice. And many of you probably know this already. People say, well, what is cancer? So basically, our cells start to lose their normal control. And Dr. Dart will talk to you a little bit about that in the second part of the session. But basically, we've got uncontrolled growth. And then eventually these cancers may spread to other tissues, and you can get cancer literally in any organ. So as an oncologist, I'm kept very busy with lots of patients coming through the system. And it is really common, unfortunately. So this is the figure now, we're quoting it in the UK, that 1 in 2 people in the UK are going to get a cancer diagnosis. But I say this all the time, it's really important to remember many of those patients are going to be cured. Okay, so it's not that 1 in 2 are going to succumb to cancer, because actually our treatments have been getting better and better. And certainly over the last 30 or 40 years, survival from a cancer diagnosis has improved significantly. And patients, if they're diagnosed early, can have curative treatment and go on and live a normal life. So our figures are a bit scary saying 1 in 2, but actually our outcomes are getting better and better. And if we think about, why are we seeing more cases of cancer? Certainly in the UK and probably globally, this is applicable. The biggest risk factor for developing cancer is age. So as you get older, your risk of developing the cancer increases. And certainly in many countries now, we've got an aging population. So as patients are getting older, their risk of cancer is increasing. So we are seeing more and more cases. But keep that within the context of how our outcomes are getting better. So even older patients who get a cancer diagnosis can do very, very well with some of our treatments. And if we think about the treatment of cancer, I've put it here, we've divided up into different modalities. So often we'll have patients who have an operation to remove their cancer, but more and more we're needing to think about systemic treatments and targeted treatments. And that's what you're going to learn about on this degree program, how cancer develops as a disease process and then potentially how we can treat it. So chemotherapy, radiotherapy, very important. And that's what I do in my day-to-day job as an oncologist. And many patients nowadays will have combinations of treatments. So we're seeing patients, then they have an operation, then they have chemotherapy, then they have radiotherapy. And something that's going to be very, very pertinent to you potentially as future cancer researchers. We are starting to look much more now at a personalised treatment for cancers. We're looking at cancer at the molecular level, and that's what you need to learn about. So do these cancers have particular things that we could target, certain genetic features and molecular features that we can target to either treat the cancer or prevent the cancer or prevent resistance from treatment? So we're really moving into a new era of precision medicine and targeted medicine for cancer, which is quite exciting because it's really moving patients forward in terms of treatment. And if we think about, what's been happening with cancer development and where do we see your role as our cancer biomedicine students? Well, my job wouldn't be possible without all the cancer research that goes on. We need to understand the science and the mechanisms of how cancer develops and how we might diagnose it. So that's the role of the cancer researchers. So finding out, why certain cancers are so prevalent, and how can we screen for cancers? It's much better to diagnose patients earlier when you can intervene. So that's a very important tool. And always thinking about new treatments. So how are our novel treatments working? Chemotherapy has been around for a long time now, and we need to have new techniques and new treatments for our patients. And one of the big problems we see in clinical practice is that our drugs stop working and patients develop resistance. And overcoming resistance to treatment is really important from a research point of view so that we can get patients successfully having treatment. And as we said at the start, I may run many clinical trials, but I wouldn't get to trials if the researchers hadn't done the groundwork for that. So discovering the mechanisms of action of drugs, what's going to be the safe dose, how do they work at the cellular level? It's really, really important. So we talk about this concept of bench to bedside, and I think it's really, really important. So the scientists working at their bench, doing all the work, doing the research, and eventually that will translate into clinical practice, and it's hopefully going to be treatment that we can give to our patients. And we work very closely together, scientists and researchers and medical teams as well. I think it's fair to say that the UCL Cancer Institute has got a worldwide reputation for cancer research. And I love showing this slide because on the left-hand side here, we've got the Cancer Institute. On the right-hand side, you've got the Macmillan Cancer Centre where we see our patients. And they're literally just across the road from each other. So we work very closely together. There's many people who have labs in the Cancer Institute who are also seeing patients in the Macmillan Centre. So it really brings that bench to bedside approach, to prominence. And it is a world leading centre for research, and we collaborate with many of the hospitals in the area, so the Royal Free Hospital, UCLH, Great Ormond Street, and you'll see firsthand how the science is eventually translated into improving outcomes for our patients. And when I say we are really world leaders in cancer research, it's absolutely true. And I've just picked a few sort of headlines from the last sort of 1 to 2 years just to show what our research is doing. So you can see things here, lung cancer vaccine trials, really pioneered by UCL, and melanoma vaccines. Charlie Swanton, who's one of our scientists in the Cancer Institute, a world leader in lung cancer. These are big names, groundbreaking sort of discoveries in terms of cancer treatment. So we big up the Cancer Institute because we really should. We're doing some fantastic work there. Some fantastic research is going on. So, why do we think you should do our course? Well, I've said, that patients are doing better, they're living longer, and probably because our treatments are getting better. And that's where we need future researchers. We need to keep driving things forward and improving outcomes for our patients. And, the research, we need to understand, well, why do patients develop cancer? How do we help them to get diagnosed early? How can we screen for cancers? How can we develop new drugs? This is all very, very important to deliver better care for our patients. So we feel this is a really relevant topic. It's a common condition. We're a internationally renowned institution, so it's going to be a very relevant, stimulating degree for our graduates, and hopefully we're going to really open the doors for them in the future to carry on innovative work in cancer research. So a little bit about the course. So just to break it down, so year one, first of all, I guess is a very general introduction to health and disease, and that is with a number of the other IMS programs, which I'll mention shortly. So you can see that you're learning sort of the basics, so biology, anatomy, biochemistry, etc. You'll get an introduction to cancer in that first year. So we have a module which I run, Cancer Medicine in Society, which is sort of trying to set the scene for the impact cancer diagnosis has on patients, the cost of cancer care, and thinking about it sort of more globally. So that's your introduction. So you get your first years, the sort of basics of health and disease. And then as you move into your year two and year three, your modules become much more cancer specific. And you can see we've listed there some of the core modules that you'll undertake as part of this degree program. So you'll be learning about, cancer biology, therapeutics, how we run trials, how we develop trials. I mentioned, consider very, very important. We are starting to individualize treatment for our patients. Radiotherapy, very, very important treatment modality. So you learn about that. If you think globally, infections account for a significant amount of cancer, so pathogens in cancer, all very relevant. The impact of our diet that can have on cancer development and our treatment. So you can see there a wide range of modules. And we also have optional modules as well, which we try to be as flexible as we can, because we appreciate some students might have other interests. So if we can make it work with the timetable and it's relevant, we would encourage that as well. And then in your final year of the BSc, you will do your research project, and you might do some lab work in that time, or you might want to do a sort of dry lab project. So that's very much open, that you have some choice in what you want to do. And some of you may be interested in the MSci. So this is allowing you to study cancer at master's level. So that would be a 4-year program. And that's quite a unique program at the moment. So we're quite proud of that. Thierry was very instrumental in bringing that to fruition. And we've got students going through the MSci program at the moment. So again, it's master's level. So you'll learn a bit more about advanced therapies, new therapies, and really get some skills about how to conduct cancer research. The MSci, you do need to be doing well in your first 3 undergraduate years, so it is based on academic performance, and there is some flexibility to transfer between the two courses. So what are you going to learn on the course? So I've just tried to summarize some of the key things that you might learn during your 3 years. So we've talked about, what is going wrong with cancer diagnosis. So why do cancer cells start to lose control? What's happening at the cellular level? What's happening with all the pathways that allows uncontrolled replication? Why are so good at surviving? Why do we get resistance to treatment? This drive to survive and this drive to spread and metastasize is very important. So why is that happening? When you think about, basic science, and, we grow cells in the lab, so you'll learn sort of very good laboratory skills, and then you can start to do those initial experiments. So, thinking about, what might affect cell growth, so you'll learn sort of cellular techniques, so cell cytometry, things like that. Very important sort of basic science skills. Infections I mentioned, so we talked about the fact that you think globally infections cause many cancers. We've got some vaccines coming through to help prevent certain cancers. Why does that happen? What's the role of different viruses and infections in causing cancer? The immune system, so important now in terms of the development of cancer and the treatment of cancer. So many of you may know about immunotherapy. So in some of our cancers, we're not even giving chemotherapy anymore. We've moved into the immunotherapy era, where we're manipulating the immune system to try and treat the cancer. Very, very important in terms of therapeutics. Chemotherapy, I said, for some patients, they get combinations of treatment. We still use a lot of chemotherapy. How does it work? What's happening at the cellular level? Why do patients become resistant to chemotherapy? Very important things to understand. Radiotherapy, also very, very important treatment for our patients. So we need to think about the physics and the science. How does radiotherapy actually work? How does it go into the DNA of that cancer cell to be a therapeutic tool? We have a module on radiotherapy that you'll hear about. Precision cancer medicine and precision cancer care. So I said, we are moving in to an era where we are much more trying to personalize treatment. So I have many patients who are now on targeted treatments because they have a particular mutation that we have a drug that can target it. So we're definitely moving to that era of much more sort of precise, individualized cancer care. And clinical trials, very, very important in my day-to-day practice. So we think about, how do trials get set up, the ethics behind that. How does a drug get to market? How is it approved? And how do you make sure a clinical trial is robust and going to give you really powerful statistics? So you'll learn about not just how we set up general trials, but specifically cancer trials. And we think, the expense of this all, I've said cancer is really common. The new drugs that are coming through are very, very expensive. How are we going to afford all of this? How do we make decisions about what drugs are available for our patients? So all of these big topics will be covered in the various modules that you do over the degree program. And I mentioned about that first year where you are with the other medical sciences degree program. So we talk the IMS program, so integrated medical science. So that would include all of these degree programs. So you can see the BSc alongside Cancer Biomedicine is in there. So in that first year, as things stand at the moment, we have our own students. But if you suddenly decide, well, I'm not sure this is for me, I might be more interested in sports science, there is that opportunity to switch. And equally, we take some students who maybe have started with applied medical science and think, actually, I'm really interested in cancer. I'm going to do the BSc in Cancer Biomedicine. So there's that option in your first year to switch within the IMS programs, which is quite a nice way that you're not absolutely 100% committed from there on. And just a few comments from our students, because I think it's important to hear the student voice in all of this. And these are some quotes that we've got from some of our surveys that we did with students. So, they like the course. We're quite a small course, so everyone tends to know everyone. And I think, our students really appreciate the fact that they're working with these world-class researchers. As a teaching team, I think we'd like to see ourselves as being supportive and approachable. And, we get a lot of good feedback from our students. They do enjoy their time with us. People think long term, so where do our students end up? So we've got quite a few years now of students graduating, and we're seeing sort of patterns emerging. So you can see some of the career destinations of our alumni now. So it's great to see many of our students are going on to stay in research, in cancer research, and they're doing their MScs or they're doing PhDs. We've got students who are keen to go into graduate entry medicine, so this would be one of the routes for graduate entry medicine, or you could work in the pharmaceutical industry, you could help run clinical trials. There's lots of different career options, and you're not forced to stay in a cancer sort of theme, because I think the skills that you learn are very applicable across many different types of biomedical sciences, so you can apply your skills elsewhere if needed. And a little bit just there to look at the entry requirements. This is all information that will be available to you through the prospectus. So you can see the offers there, and the IB payment levels as well. That will be available in the prospectus. So I'm going to hand over now to Alwyn, and then after Alwyn's session, we can have a Q&A. So I'll just stop sharing my screen and let Alwyn step in now. Thanks, everyone.
Let me try and... Can you let me know if that's coming through okay?
Yeah, I can see it.
Yeah.
Perfect.
Okay. So bear with me one second. So, okay, thank you. My name is Alwyn Dart. I'm just going to give you a little taster session of the kind of lectures we have in our second year when we kind of dig into what goes wrong in cells for them to become cancerous. So in today's taster session, I'll start off by asking you very quickly, just to think for a few seconds, what words come to mind when you hear the words cancer or cancer cells in terms of their characteristics. So just take a couple of seconds to have a think. Some of the clues are actually on the screen in the picture. So when we think about, oh, Bear with me. There we go. When we think about cancer cell characteristics, some of the main things that we deal with or we think about are they're a population of rapidly dividing cells. They're dividing out of control, and they have the capacity to become motile. They can spread across the body in the process called metastasis. So when we look at the tissues, the normal tissues in the body, for example, here we have an example of the large intestine and the prostate gland. If we were to take a cross section through a small area of those tissues, we see that that tissue is made up of a highly structured and ordered population of cells. So you can see here that in the large intestine, we have the villi, and then the surrounding underneath tissues are the muscle and the blood vessels. And in the prostate gland, we have glandular epithelial cells lining ducts within that tissue. So we know we have a tightly regulated population of cells and structure. So in these tissues, cells can grow and divide. They can perhaps go into a resting state. They can differentiate into other cell types, or they can undergo a process of cell death called apoptosis. So how does this well-ordered cellular structure become deregulated in cancer? So again, for a few seconds, just imagine that you were a cell within a tissue like this. What questions would you need to ask yourself before you decided to divide? So take a couple of seconds just to think, what questions would that cell need to ask before it decides to divide? So the two main questions can be divided like this. The first question is, should I divide? So under the heading of should, the cell needs to determine whether it's appropriate. Is there anything telling that cell that it should divide? Is it required? Does the overall tissue require more cells? Perhaps the tissue is undergoing some form of stress and more cells are actually are required. And also cells need to determine is everything okay before they start to divide? Is there any damage in the cell that needs to be dealt with before they pass that on to the next generation? And the second kind of pathway of decisions the cell has to make is, can I divide? So does the cell have enough energy? So the cell division process is a highly energy dependent process. So does the cell have enough energy to divide? And does it have enough nutrients to divide? So in terms of nutrients, we mean the lipids, the DNA, and the proteins that are all necessary to produce an entire new daughter cells. So these questions all need to be asked and answered within the cell for it to determine whether it's going to undergo cell division. So we know that cells are aware of their surroundings and they respond to their surroundings, and they're in a kind of constant communications, either with other cells in the vicinity or with molecules that pass outside the cells. So the cells use all manner of communication systems. For example, they use protein hormones, growth factors, cytokines, steroids, and in some instances, some gases as well. And all these molecules are detected by receptor proteins that are located for the majority on the surface of the cell. So they have a compartment that protrudes outwards to detect these passing molecules, and an internal part of the protein that signals into the cell, which I'll explain in a moment. So if we were to zoom in on the surface of any given cell in the body, we would see that it's coated with a large number of different receptors for each of these communication molecules that the cell encounters. So there's a separate receptor for every type of signaling hormone. So if we were to examine how these work in detail, for example, outside the cell, we may have a circulating hormone, for example, which might bind to one of these receptors. When this happens, the receptor is changed in a way that turns it on, so it's switched on. This receptor is now switched on and signals intracellularly inside the cell to various adapter proteins that carry that signal forward in a cascade, which eventually results in perhaps a response from the cell, which could be a growth response or a gene activation response. So these cells signal to each other and they respond to external stimuli, and quite often the response is cell growth or cell division. So cells are in constant communications with their surroundings and each other. They respond to growth stimulatory signals, growth inhibitory signals. They respond to nutrient levels, and they're also monitoring if they've got any DNA damage that they need to deal with. If the answer to the questions, is it appropriate and can they divide and is everything okay, is yes, then the cell will divide. So the process of cell division is controlled by a whole plethora of different genes, two categories of which we're going to discuss today. The first one is oncogenes or proto-oncogenes. These are normally genes in the cell that have a growth stimulatory function. For example, they could be the growth hormone receptors. As we saw on the previous slides, these act to stimulate the cells to respond to the growth stimulus. So they can act as accelerators. If these genes acquire mutations that make them more active, or they can become duplicated or overexpressed, this can increase this growth potential and they can act as accelerators on the cell cycle. So, for example, if we had a mutation in one of these receptors, and this receptor was already in a permanently switched on position, even now without any external stimulus, this would then also trigger to the downstream signaling proteins, and this would lead to a response from the cell. But this time, as we can see, the response is inappropriate because there wasn't an external stimulus that triggered this pathway. So these cells are now in a permanently switched on position. Also, in a very similar manner, if we were to have an overexpression of one of those receptors, then you can see that they would then amplify any external stimulus and again filter down through the cell pathways and cause an increased response in the cells. So we have an increased level of cell growth or cell division. So I'm going to use a very old analogy, but it often proves to be quite useful in trying to picture what's going on. So if you were driving a car, for example, you have two ways of controlling that car's velocity. You can press the accelerator pedal down to make it go faster, or you can press the brake pedal to slow it down. And we have very similar mechanisms in our cells. So for example, if one of those oncogenes, one of those receptor genes, becomes mutated and becomes permanently switched on, this is analogous to a car having its accelerator pedal permanently pressed down. So you can see that that car's velocity is going to increase. However, as in the car, we have brake pedals. So if things start to get out of control, the brake pedal can be pressed to reduce the velocity. Cells have a very similar set of proteins within them that can act as brake pedals on the process of cell growth and division. And also these brake pedals help to stop the cell cycle if there's any repair that needs to be undertaken for the DNA, for example. So in our normal cells, our DNA is constantly being attacked from things in our environment. Even simple things such as oxygen and radiation from our natural environment is enough to cause quite a high frequency of DNA damage or DNA breaks in our normal cells. But normally in these cells, the cells would stop their cell cycle, they would press that brake pedal, stop the cell cycle, and give that time for that cell to repair the DNA damage, and then it would continue on its way. But if the cells acquire mutations in those stop signals, if they can't press that brake pedal to halt the cell cycle and give themselves time to repair the DNA, then the cell will continue to divide with DNA damage. And DNA damage that is inherited from one cell to the daughter cell is when we start to get mutations in genes. So you can see how this then starts to spiral out of control. If we then have more mutations and they affect things like the oncogenes, those accelerator pedals, you can see how things start to spiral out of control. So in a normal cell, they would repair their DNA and then they would continue with their normal cell cycle as healthy cells. But in cells with defective proteins to stop the cell cycle or defective DNA repair enzymes, the DNA damage would persist and the cells would then continue to grow with the damage with mutations in their genes. And this can then start to lead to cells becoming abnormal, which gives them a kind of start onto the process of cancer. So this also is an interesting sidestep. So some of those genes involved in the brakes of the cell cycle, the tumor suppressor genes, they're quite often destroyed by virus infections. So, for example, in cervical cancer, the HPV virus infects the cells, and the proteins encoded by the virus itself can destroy some of those proteins that act as brake pedals. So those cells that have become infected can no longer stop their cell cycle to allow time for DNA repair. So this is an interesting aside as to why some vaccination programs, for example, HPV, can reduce the kind of rates of certain types of cancer. So if we go back to my car analogy again, if we have, for example, a mutation or a loss of these brake pedals, then that car is going to start to accelerate out of control. And obviously, the worst case scenario is what would happen in this analogy if the accelerator pedal was stuck down, but also if we didn't have any brakes, then obviously that car is going to accelerate out of control. And this is exactly what happens within the early cancer cells, where they have a loss of a tumor suppressor gene, so they can't stop their cell cycle and they can't repair their DNA. And they have a mutated oncogene which acts as an accelerator, which presses down and causes the cells to grow faster. So when we have faster growth and no cell cycle arrest and no cell cycle stop, then this leads to a rapidly dividing population of cells. Obviously, this isn't quite enough to become the disease we know as the kind of life-threatening disease we know as cancer, but it gives you an indication of how a small population of cells can start to lose control over their cell division cycle, and they can then start to to grow out of control. So the tumor suppressor genes tend to hold the cell cycle back and act as brakes, and the oncogenes tend to drive the cell cycle forward. But there's multiple mutations that need to occur in both the oncogenes and the tumor suppressor genes for the full disease we know as cancer to occur. So then we refer to cancer as being a multi-step genetic disease. So how can you tackle cancer, the cancer burden by studying cancer biomedicine? So we know that if we can study the causes and understand the kind of molecular mechanisms behind many cancers, then we have a better appreciation of how to develop drugs and therapies for cancer. In the next 10 to 20 years, there are likely to be some groundbreaking therapies in cancer, for example, the immunotherapies. And as students on a biomedical cancer biomedicine course, you will become the cancer researchers of the future to make discoveries and to save lives. So I'm going to finish off there and just leave you with the question slide, and I will stop sharing and get back into the meeting. So bear with me 1 second. There we go. Okay. Is that okay? Is that coming through? Yeah. Okay. Okay. So did we have any questions in the Q&A?
Okay. So someone asked, I'm interested in studying biomedical sciences or infection and disease or cancer biomedicine, but I'm worried about finding work after studying at university in London and abroad. Any advice?
I mean, I'm happy to try and answer that one because I think hopefully I covered this a little bit in my slides about future careers. So, certainly from our students, there's still a lot of opportunities. You learn about infections, you learn about other diseases. So I think we're not finding that our graduates are having difficulty getting jobs in the future. So a UCL degree is it's got a very good reputation with it. So I think our experience, I think that's fair to say, everyone, yeah, that our alumni, our students go on to do very well finding work after their studies.
Okay. Someone also asked, how does a biomedical course at UCL differ from a medical course? So I
I think it's important to recognize, medicine is a completely separate degree. When you do medicine, you're training to be a doctor and work as a doctor. That's not what our graduates are going to be doing from this point of view. So this is very much more a science sort of background. And so we're thinking more, our graduates are thinking about staying into cancer research. Now, yes, some will go on to do medicine, but it is a completely separate degree. So there's some overlap, but this is not a degree that leaves you with a qualification allowing you to be a medical doctor.
Okay, so someone asked, is there a lot of lab work in this course? I think I can answer based on my experience. So we have a lab module, and this is mostly during your first term. And so for the first year, you study a lot of like basic lab techniques. But for your first year of the course, that will only take up like your first semester. So I would say that you do lab work, but it doesn't like you don't do it throughout the whole academic year. I hope that
I,
answers your
I
question.
I could add some to that as well. As we progress to year two, there is a module dedicated for techniques in molecular medicine. So you'll have a series of lab skills again. And then in your third year, again, you will do some labs as in a more focus on cancer research and obviously your research project. If you're going to go on the program, MSci program, and again, that will entail quite a lot of lab skills. Again, we have a dedicated lab skill module in the fourth year, and it's also leading into a master's level project. So you're going to have, if you're joining on the MSci program and you're going to carry on with the MSci program, you're going to have significant level of lab work and research skills developed through. That will pave the way for you if you want to be the future of the cancer research.
Okay, another question is, how are the exams and coursework? Is it too much? Okay, so based on my first year, I would say that it is definitely different to what you're used to, whether you're studying A levels or I don't know, IB, but the amount of time that you're given to complete your coursework is sufficient if you're smart with how you manage your time. And for the first year, I think that you do a lot of group work. So most of the coursework that you're given in the first year, it just depends on how you interact with the other students. So if that's in your group. And for independent coursework, you have a lot of ample time. Usually your due date will be like a month from when it's assigned. So I think if you're worried about whether it's too heavy, as long as you keep on top of it, you should be all right. And for exams, for first year, you only have exams during May, June. At least for me this year, this is like not it doesn't clash too much with your coursework, but we do have something called a capstone, and that runs like during the end of May to June. And that kind of like I wouldn't say clashes with your exams, but it is during your exam period. However, I think it's not too demanding. Just you need to know how to manage your time. Yeah. Okay. Another question is the master's level project for cancer biomedicine different, or is it similar to the BSc one?
I could address this because if you're going to go on to, is a very, as Dr. McGovern mentioned earlier on, we have developed a unique program. So if you're going to be joining on the MSci program, and then we have a different project in your third year, it's called structured project. So that will train you to get the necessary skills in order for you to do an extended project at master's level. So both of them will involve cancer research. But if you're going to be on the BSc pathway, there'll be cancer research project. So if you're on the MSci pathway, your third year project is going to be different. And then the fourth year will be extended time. And what we could say about projects in your standard BSc year or year 3 of your MSci year. It will be delivered as part of the term 2 of the academic year, and that would be, um, uh, concurrently delivered when you're doing your academic studies on other taught programs, taught modules as well. But when it comes to the master's level project, it comes with higher credit level as well, master's level. So it will have a dedicated full-time period of 3 months to conduct that project. So that will be a significant part of the MSAR year. So 50% of the fourth year will be taught, and 50% of the fourth year will be research, and that's dedicated to your research project. So it's an extended version of that. It will have the breadth and depth in that project in the final year.
Okay, a follow-up to the research project. Someone's asking if there is guidance with the final research project or if
As a
that's independent. Oh, sorry, go ahead.
Thank you. So as a cancer biomedicine student, you will be allocated, we will offer, you'll aim to offer a project based in cancer. So that's something, but all the guidance will be as you join, throughout your academic years, second year on, midway point of your second year, we start telling you about how the projects work. But then we do provide clear guidance on what projects are available, how projects are allocated. Students do have an opportunity to choose and rank projects, and then projects that get allocated based on that process. So you will have well-informed information regarding projects when you reach to that level of considering project for your year 3, subsequently in year 4.
Okay, so we have a few questions in relation to the taster lecture. One of them is there any way to treat cancer by repairing mutated oncogenes or tumor suppressor genes?
I would say theoretically, yes. In cells that we grow in culture, that can be done. There is the technology to do that and to repair DNA mutations. To do it on the scale of a patient with a large tumor is currently, I think, not available. But theoretically, you could do that. But I've just mentioned the 2 different genes, the oncogene and the tumor suppressor genes. In a really aggressive tumor, there could be tens or 20s or even hundreds of different genes that have become mutated or differentially expressed. So that would be a very challenging thing to do. So theoretically possible, practically possibly not at the moment.
Okay. And also what can be done when cancer is becoming resistant to existing treatments over time?
That is the ultimate question of cancer research.
That is also
Yeah, so a lot of the therapies that people use initially start off as very successful and very useful, and they can prolong a patient's life considerably. But at some point, the cells adapt and fight back in a way, and they either have new mutations or new gene changes that allows them to escape that therapy. So basically, that process then starts again. So there needs to be an increase then in research to find out what's changed and to kind of match a new therapy to that new molecular change. And there's a constant evolution and a constant chasing of the cancer cells to kind of by people who develop the therapies. So I don't know whether you need to add anything clinically.
No, you're right. I mean, it's that's that's the big conundrum. Yeah, yeah.
Okay.
okay. So in relation to because we offer a BSc and an MSci, someone's wondering if you have to decide between the BSc and MSci from the moment you apply, or if this is a decision you make throughout the course.
I could address that. I think at the moment, it's MSci is offered outright. So if you enroll under the MSci and then you want to consider based on, as we mentioned about academic progression, and you can continue with that. So that's a priority given for students coming in directly on the MSci. But MSci places are very limited for the fourth year, and it's very tailor-made. So if you want to think about making a decision to join the considering a 4-year integrated master's, you could join outright on the MSci, but you still have the opportunity at the end of third year if you don't want to continue with that. So, but others as well, there will be opportunities to consider an MSci in your year 2. Then you can express your interest, but that's subject to, again, how many places and so on. So again, I think look into those prospects and what do you really want to consider, whether you want to consider it a potential research career. But MSci will come with it's taught, although it's master's level qualification, it comes with a lot of benefit as well. So you do have it's delivered over 2 terms or exams and assessments in term 3, but compared to a full-time master's, which will run for a full academic year. So you're looking at a possibility of thinking an MSci. It works together, not as that additional year, which is also what we teach over the first 3 years. So it integrates into it. So as a whole, as a degree, 4 years, you have to look at that way to gain that whatever you need to gain out of the degree. So there will be opportunities to discuss with us about the MSci if you're considering, even if you join on the BScs when you join for BSc. So you can consider that opportunity again.
So I think a follow-up to that is how many people are typically on this cancer biomedicine course and how many people are on the MSci?
So we typically aiming to get around 40 students, and then usually we think about 10 MSci students to progress to the fourth year, because we want to enrich that curriculum with a more personalized experience for a student in the fourth year. So think about carefully how, are you going to be interested in cancer research? And then MSci would give you the necessary skills to bridge the gap if you want to consider doing a doctoral degree beyond your degree. And I think the MSci Cancer Biomedicine provides that necessary stepping stones in terms of embarking on that career. It could change over time, but we are flexible. But we like to keep the programs quite small to give that small group level learning, interactive learning. So yeah. So at the moment, as it stands, about 40 in total.
Okay, I have a question. There's a question that's asking, what are some examples of the optional modules mentioned for year 2? Okay, so for year 2, the optional modules are offered by, like, they're run by a few different departments, if I'm not wrong. So, for example, for me, when I applied for my optional modules, I chose Cancer and Nutrition, which is under the Cancer Institute. But there are also optional modules like Applied Tissue Engineering and Immunology, and there's even things like Science and Ethics. So there's a range of different optional modules that you can choose. For year 2, you can choose up to 2. And those are kind of like to broaden your learning, and they're kind of just a way for you to learn things that you're more interested in alongside the compulsory ones for your course. Yeah. Another question is, if you begin the BSc course and change to the MSci, at what point during your time at uni would this take place?
Process will because we do a different the year 3 is exactly same as the BSc for the MSci except the project. So if someone considering switching to the MSci, then that will be considered in the second term of year 2 academic year. So and then it's a process of going through that process. But yes, you can switch to that point, and then third year you do a different project instead of doing a standard a research project as part of the BSc program. But the MSci will have a different project called structured project. But however, in both scenarios, the students have to achieve an academic performance. You know, there is a requirement to get that academic performance in order to progress to the MSci. Even if you're enrolled on an MSci year, you still need to meet that requirement.
Okay. So I will take like three more questions before our session ends. So one of them is, does our course focus on any particular types of cancer?
I mean, I can answer that one because I think what we try and do is cover, most of the cancers. So, for example, in year two, the clinical cancer module, you'll learn about all the common cancers, and that's usually taught by oncologists. I think one of the questions was, do I still work do I see patients? I absolutely do. So a lot of the teachers on the course are working, doctors as well. So we tend to cover all of the common cancers. You'll do some hematology, and then potentially in your project you may really specifically focus on one particular cancer type. So I think we give a very good generic sort of overview of all the common malignancies.
Okay. Another question is, I think this is in relation to the lecture from earlier. Do you learn, oh, sorry, no, this is just in general. Do you learn about gene editing in this course, for example, CRISPR?
Yep, I think that would be in year two, wouldn't it, if they were in the skills module where they would learn some basic molecular biology techniques. Um,
Yes, you'll do that. It's a module, as I said, dedicated to molecular biology, practical skills. So there'll be content on that.
Okay. Okay, are there any upcoming open days to attend in the future? I believe for the integrated medical sciences, there's an open day in September, but is there one specific to cancer biomedicine?
We do have in-person open days in June. And also, as you said, that we do have integrated medical sciences where cancer will be represented as well in September.
Okay. A few more questions. Okay, this one is in relation to the lecture. How do our current cancer-directed technologies incorporate cell proliferation into their design?
So,
Oh,
I think if you're talking about therapies that are directly targeting cell proliferation, I would probably think that historically the kind of the nastier chemotherapy agents would have been the ones that would be targeting just purely replicating cells. But I think that with the kind of more modern precision therapies, they target some of those receptors that I mentioned in my talk. They are directly targeting those receptors that are permanently switched on and mutated as well, importantly. So they can be much more precise, so we can avoid some of the more kind of generic side effects you see with things like chemotherapy for specifically for cells that are rapidly dividing. So I think the more modern view or the future-facing view is to try and engage therapies that don't just target that as being the main characteristic. So it's not just because the cells are replicating fast means that that's how they should be targeted. So the kind of more future-facing therapies are the ones that are going to target those specific mutations that I mentioned in those receptors or the oncogenes, and to try and engage the immune system, I think Ursula mentioned as well. So those are kind of, you could say that the whole focus is shifting away from cell division and replication, and that's it. Hopefully, in the future, therapies will become much more tolerable and with less side effects. I think that would be the way forward. I'm not sure if that answered the exact question. But I think that's the way we look at it these days. Yeah.
Okay, and the last question, I can answer this. How many compulsory modules do you have to do in year two? So for the compulsory modules, you have 6, and then option modules, you'll have 2 for your second year. Okay, so I think that's all the time we have for today. So I'll leave it there. However, if you could please provide us with feedback about today's session by filling out our survey, which will appear on the browser at the end of this event, it would be greatly appreciated. Thank you for all your comments and questions, and thank you very much to our speakers for an excellent session. Have a great evening, everyone.